So again, if you're just tuning in, welcome to What's New in MPMI. I'm Jean Harris. I'm the editor-in-chief of MPMI Journal. And I've really been enjoying this series as a way to share with you some of the new research and writing that's taking place in the journal, to share it with you from the authors themselves in real time. And this simultaneously is overlapping with another big effort of MPMI, which is our top 10 series. What are the top 10 unanswered questions in MPMI? And so the last time we met, I gave an overview of the whole series, talking about introducing the 10 questions. And today we're going to have the first review that came out in the November issue of MPMI. You can find it, I think that Ashley is going to post that in the chat. So please take a look there to see how you can access the list of questions and also the paper itself. And so delighted to have Ralph Panstruga and Matthew Moscou from, Ralph is from Aachen University. Matthew Moscou is from the Sainsbury Lab. And they're going to be presenting the first in our series of the top 10. And this one is a fascinating question people have been pondering for years, and also a lot of things we still don't know, despite all of this work. And so I'm going to turn it over now to Ralph and Matthew. Remember, if you have questions, please type them in the Q&A box, and we will get to as many as we can at the end. Okay, thank you.
Hello, everyone. Happy New Year to all of you, and many thanks. I'm really, stunned by so many people watching our presentation. I hope you had a good start into the new year. And today, as Jean just announced, we will present the review that was recently published about non-host resistance in MPMI. So I will start sharing my screen so that we all hopefully can see my slides. And yeah, this is the article we report today. It's a bit weird for us to report on a review article instead of an original research paper. But well, there might be a special reason for that. And this is because this is one of the papers of this top 10 series of unanswered questions in molecular plant microbe interactions. I will come back to this in a second. So this little cartoon here is an illustration of the two of us. On the right hand, it's Matt. He looks a little bit different right now with his beard. And on the left hand, it's supposed to be me. And all these nice illustrations have been drawn by Matt's wife, by the way, for those of you who don't know. All right. To give you a little recap on what this top 10 series is about, probably most of you know, but some of you might not know. I've introduced this slide here. So at the IS-MPMI Congress in Glasgow in 2019, participants were asked to write down what they think are the most important questions in the area of molecular plant microbe interactions. And then many suggestions were collected, and they were counted, and then they were ranked according to their how often these points were raised. And then this top 10 list came out in the end, and you can see here the board at the meeting where we could post our suggestions for the top 10 questions. And one of these questions, and it's actually the question number 6 highlighted here by this arrow, was what's the molecular basis of non-host resistance? And in the following few months, we got in contact with Jean, or she got in contact with us, and we agreed to write a review on this topic. So why why us? So this might be explained on the next 2 slides, which give you a brief introduction to us. So first, a few few data on Matt. You can see here on the photo and on the right on the cartoon again. Matt is a group leader at the Sainsbury lab in Norwich in the UK, and his research focus is really on non-host resistance. So he is really the expert on non-host resistance. He's trying to understand the genetic basis of this phenomenon and try to map and clone genes that are involved in non-host resistance. And for this, he uses both natural variation as well as induced variation, so mutagenesis in non-host species, to identify the underlying genes. And he does this in the context of various plant diseases, and his particular pet diseases are the cereal rusts, mildews, and blast diseases. And he uses on the plant side various grass species, including barley and also Brachypodium distachyon. And by the way, I just learned that the common name for Brachypodium is purple false brome, which I also didn't know until now. Well, a few words on myself. So I work in Aachen in Germany. I'm professor in plant molecular cell biology. My research focus is on plant powdery mildew interactions at the genetic, cellular, and molecular level. And one of my research interests is so-called MLO-based broad-spectrum powdery mildew resistance. And this type of immunity shares many characteristics with non-host resistance to powdery mildew fungi. And that is why I am also interested in non-host resistance. Well, for those of you who can't spend the whole time with us today, I give at the beginning already a summary of the main points we would like to make in this presentation. So this reflects a summary of our review article. As a first aspect, I would like to stress that we think that non-host resistance is not a mechanistic, but a phenomenological term, which is often confused. So people, when they think about non-host resistance, they think this is a particular process in which specific genes are involved, which are only there to mediate non-host resistance, and they do nothing else. But this is not the case. In fact, non-host resistance is just a phenomenon, and based on this description of the outcome of the plant microbe interaction of the resistance phenotype, it is really defined and not on the basis of the underlying genes. And we will come back to this later. Then we would like to stress that non-host resistance comprises many more facets than usually considered. So many people think of non-host resistance in terms of resistance and susceptibility, and that's it. But we will point out that it is not that easy. There's not only black and white, but there are many shades of gray in between, and many additional things that influence the outcome of the plant microbe interactions, many additional features. Then we will point out that non-host resistance relies on the very same genes and pathways as other types of plant immunity. We think this is very important to stress. And therefore, this term non-host gene, which is often used in the context of non-host resistance, describing a gene that apparently has a role in mediating this type of immunity, is very misleading, because it implies that this gene really might have a specific function only for non-host resistance, which is, in our view, not the case, since in to the best of our knowledge, all of the genes that have been identified so far that play a role in non-host resistance also play a role in other types of plant immunity. Non-host resistance also involves the recognition of pathogen effectors by host immune sensor proteins, at least in particular settings. And these immune sensor proteins are, for example, the famous NB-LRR proteins. And later, Matt will tell you more about this. This might then also give rise to host shifts and host range expansions, because there are gains and losses in the course of evolution in the armories of both the host and the pathogen, and then these gains and losses of effector proteins on the pathogen side and immune sensor proteins on the host side may then also lead to changes in the host range. Thus, the extent of non-host resistance defines pathogens' host ranges or contributes to the definition of pathogen host ranges. And as Matt will also demonstrate to you, in some instances, it is possible to transfer a gene that contributes to non-host resistance in one plant species to another plant species, and thereby boost immunity in the second plant species, resulting in a broadened disease resistance or maybe a broader range of non-host resistance. Well, these are the most important points we would like to cover in this presentation. And I will start off very easily to explain what is non-host resistance. And when we look into literature, we find that non-host resistance in the past has often defined as the ability of all genotypes of a plant species to confer resistance to all genotypes of a pathogen species, where resistance was then often considered the inability of a pathogen to complete its asexual or sexual life cycle on that plant species. And I would like that you keep that in mind for maybe 2 minutes or so, because I will come back to this aspect of all genotypes of a plant species and all genotypes of a pathogen species on the next slide. So please keep that in mind. But to illustrate the basic concept to you, I've brought here an illustration, and this illustration refers to my pet pathogen, Plumeria graminis, forma specialis horrii, which is an ascomycete fungus and the causing agent of the powdery mildew disease in barley. And when spores of this pathogen land on a barley plant, you can see that the pathogen can infect the plant, it can grow, it can form colonies, it can sporulate on this plant. However, when the same spores of this fungus land on an Arabidopsis plant, you can see nothing. And this is due to the fact that on this plant, Arabidopsis, fungal pathogenesis is arrested very early. So the fungus cannot penetrate into the host cells. It cannot form its feeding structure, the so-called hostorium. There's no hyphal growth, there's no sporulation, no colony formation, and therefore we can't see macroscopically anything. While we see these fungal colonies here in this whitish color, you can see nothing here in this case. So, and this is then considered to be a classical non-host interaction. So we call the pathogen, when it grows on its host plant, an adapted pathogen. We call it a non-adapted pathogen when it tries to infect a plant where it is not adapted to, and this plant is then considered to be a non-host plant. So, and the topic now we would like to discuss is why is it, or what are the immune responses that are active in such a non-host plant that repel the attacks of this pathogen, while apparently this is not effective here in this setting in the barley plant. So I come back to what I showed you on the previous slides, this definition of all genotypes of a plant species against all genotypes of a pathogen species. There are potential problems of this definition when you have a closer look at it. One problem is that it is virtually impossible to test all existing genetic variants of a given plant species against all existing genotypes of a pathogen species. So based on this definition, we could never claim that a given host microbe interaction is a non-host interaction, because it is simply impossible to test all these constellations of all the genetic variants that are around in the wild. A second problem is that the definition assumes black and white scenarios. So with resistance and susceptibility being the only outcomes. So resistance defined as the absence of macroscopically visible disease symptoms, and susceptibility defined as the presence of macroscopically visible disease symptoms. But in reality, the situation is much more complicated, and we can get these grayish scenarios where there's a little bit of pathogen growth. And I will come back to this on one of the following slides in more detail. Another problem is that this definition is only true for a given point in time, namely now, as we observe it, as we describe it, as we look at it. And this is only true for maybe a few decades, centuries, or maximum millennia ago. But we don't know how this plant pathogen constellation was at, let's say, 1 million years ago, or how it will look in 1 million years from now. So there it might, due to evolution, have a different outcome. Then completion of the life cycle, as also mentioned in this definition, is a poor criterion to categorize the pathogenic success of some of the intruders, for example, bacteria or insects. For filamentous pathogens such as fungi or mycetes, completion of the life cycle is clearly defined, but what is completion of the life cycle for a bacterium or for an insect? So there, this definition fails. Then the definition also neglects that non-host resistance can be conditional, and I will also give you later some examples of conditions that affect the outcome of a plant-microbe interaction in the context of non-host resistance. So let's first come back to this gray zone. This is depicted here in this figure, which is also a figure of our review article, or as a central part of it at least. And what you can see here is, graduate outcome of plant microbe interactions from non-host scenario, so where we have complete resistance, to a compatible interaction here on the right-hand side with the host plant, where the pathogen, here depicted by the yellow spots, can nicely colonize this plant. And we have all intermediate outcomes here that are possible. So we can also have an intermediate host or an intermediate non-host where we have gradually less colonization success of the pathogen here. And this is indicated by this blue triangle. So from the left, where we have 0 plant disease symptoms and 0 pathogen reproductive success, to here in the host scenario, 100% reproductive success and plant disease symptoms. And this is also illustrated for you here with some pictures of plant powdery mildew interactions or barley powdery mildew interactions. Here we have fully susceptible plants, which represents this scenario here. And on the other end, we have here a non-host interaction with another cereal species where we can't see any disease symptoms reflecting this situation here. And intermediate outcomes are possible. We have here an intermediate host, which allows some fungal colonies to form. And then we have the intermediate non-host scenario here depicted by this photo, where when you very carefully look, you may recognize here or there a little bit of fungal growth. And actually, you have to use a microscope and look under the microscope to see these small colonies giving rise to a little bit of sporulation here. And we have put here double-sided arrows in light blue to indicate that a shift from one scenario to the other is possible. And this might be also influenced by conditions from the outside. We'll come back to this later. By the way, I forgot to mention that we subdivided our review article similar to the top 10 questions in plant microbe interactions, also into 10 questions that we addressed in this article. And this here is question 1, Q1, what is non-host resistance? So this is simply providing the background, what is non-host resistance about? What are we talking about? Our second question that we addressed in the review article was, how can non-host resistance be integrated into the current molecular concepts of plant immunity? You may know that the principles of plant immunity are generally valid. And the current belief is that there are 2 main layers of plant immunity. One is induced by so-called PAMPs or MAMPs, pathogen or microbe-associated molecular patterns, which trigger an immune response, which is called PTI, pattern-triggered immunity. And then there's a second level, And this level is induced by effector proteins secreted by various pathogens. And these effector proteins are recognized by particular immune sensors. And as promised, Matt will talk more about this. This leads to the activation of a type of immunity which is called ETI, effector-triggered immunity. And this general principle of the plant immune system, first discovered, of course, in Arabidopsis, has now been validated in many, many different plant species, and it is thought that it is generally valid. And all phenomenologically defined types of plant immunity, such as basal defense or quantitative resistance or systemic acquired resistance or race-specific resistance, rely on these molecular principles here. So these terms here describe on a descriptive basis different types of plant microbe constellations and the outcome of the resistance phenotype that we have, whether it is quantitative or whether it's systemic resistance response. All of these rely on the very same basic molecular armory that we find in the plants. And we argue that there is no special mechanism in place for non-host resistance. Also, non-host resistance relies on these two main layers of immunity that we find in all the plant species. Well, this picture here is a very simplified version of the plant immune system. On the next slide, I show you a little bit more elaborate version. We can find, nevertheless, back the two layers I just mentioned, effector recognition and effector-triggered immunity here on the left, and MUMP recognition and MUMP-triggered immunity here on the right. The difference to the previous version is you see many more players. They've got here dedicated names, and the contribution of phytohormone pathways is also put here into this scheme. But yeah, we argue that also non-host resistance relies essentially here on these molecular components that you can see here. Next, we come to question number 3. How durable is non-host resistance? Non-host resistance is generally considered to be very durable, and it is this durability of non-host resistance that sparked a lot of interest in this type of resistance, because people thought if it is so durable, well, if we could transfer it then from one plant species to another plant species, we could also induce durable immunity in maybe another plant species. But we have to admit that this durability we are always talking about is only based on experience from nature and agriculture at historical timescales. So we can only talk about decades to centuries and possibly millennia. However, we also know that host range extensions and host jumps are at least not infrequent at historical timeframes. When you look into the literature, you find many examples, many reported examples of such host range extensions or even host jumps where a pathogen species that formerly could not infect a given plant species all of a sudden can do so. So this indicates that non-host resistance might not be as durable as we usually think about it, especially not if we think in larger timescales, evolutionary timescales. And therefore, it can be assumed that some interactions, at least, that are nowadays compatible, so giving rise to disease, have been incompatible in the past or might become incompatible again in the future. Or vice versa, interactions that nowadays are incompatible might become compatible in the future or might have been compatible in the past. So there's a lot of dynamic in the system, and things may change, and non-host resistance might be at the end, if you think about longer timescales, not as durable as we assume. This dynamic that anyway exists due to evolution might gain additional speed due to human activities, such as the worldwide dispersal of pathogens, so with the airplanes and the traveling activities of human beings, we bring pathogens from one continent to another. Also, climate change contributes to speeding up evolutionary processes possibly, and the emergence of pathogen hybrids is one of additional events that further speeds up this process. And also the natural or artificial generation of new hybrid plant species. And one popular example of this is cereal triticale, which has been generated as a crossing or hybrid of wheat and rye at the end of the 1960s. And for quite some time did not have, for example, a powdery mildew disease. But at some point, then it popped up, and it turned out that also this new powdery mildew pathogen is then a hybrid of the original wheat powdery mildew hybrid and the rye powdery mildew hybrid. So if you introduce new plant hybrids, you also probably get new pathogen hybrids at the end. And natural plant populations in so-called hybrid zones, so where natural plant hybrids form, may in fact act as springboards for pathogens to adapt to new hosts. So all these aspects may affect or are likely to affect the durability of non-host resistance. Well, our question number 4 is non-host resistance constitutive, so present all the time, or can it also be conditional, stage or tissue specific, so only occur under particular conditions? And in fact, when we browsed the literature, we found that there are many documented examples for non-host resistance, or at least the extent of non-host resistance, being dependent on either the circadian clock, photoperiod, humidity, temperature, developmental stage, or tissue context. So these different parameters here all can influence the extent of non-host resistance. And we put this in our figure, the central part of which you already know, here above and below this central part, indicating that circadian clock, photoperiod, humidity, temperature, tissue context, and developmental stage can contribute to these light blue arrows here and shift an interaction from this outcome, let's say, to this one, or from this outcome to this one, or vice versa. A particularly interesting case in this context is the alternate host of the rust fungus Puccinia graminis. This rust fungus produces during its life cycle different types of spores, and it also has 2 different host plants. It has this, a cereal host, and it also has an alternate dicotyledonous host. And the fungus switches in its life cycle between these 2 hosts. And for example, the asexually generated uridinio spores lead to successful colonization of the grass host, but they fail to colonize the alternate host, the dicotyledonous plant barberry, which then in the end is a typical case of non-host resistance. So while in principle this fungus Puccinia graminis can colonize barberry, these particular spores here can't do it. It needs another type of spores, which then most likely have a different transcriptional program expressing different effector proteins, for example. Well, our question number 5 was, how does non-host resistance work? And for that, I would like to come back to an illustration from a publication by Hans Tordahl Christensen, published in the context of a review about non-host resistance in the year 2003, which nicely illustrates that pathogenic success of a pathogen depends on multiple steps that the pathogen has to pass to become, at the end, a successful pathogen and to cause disease. And these steps are mentioned here, and they comprise differentiation, they comprise the adaptation to preformed barriers, the adaptation to ancient inductive barriers, nutrient uptake, the avoidance of multiple recognitions, and the avoidance of specific recognition. And each of these steps can fail and can then give rise to non-host resistance. So we see this is a multi-layered process here. The fungus or the pathogen has to pass, and at each level it can fail. And accordingly, multiple molecular components are involved here that could be identified as a component that is important for non-host resistance. And this has been tried to figure out in the past based on, for example, genetic screens or pharmacological screens. And the outcome of this was, of these approaches, was that pharmacological and genetic screens in the non-host plants have led to the identification of loss of function in genes that are important for non-host resistance. So either by knocking out protein function based on drugs, so this is a pharmacological approach, or by knocking out a gene based on mutation, this is a genetic approach, genes were identified that play a role in non-host resistance. This was one, or were two of the approaches that were taken. A third one was the ectopic expression of candidate genes, mainly conducted in heterologous plant species. And here, of course, Nicotiana bentamiana was often used. And this is a gain of function approach. So it was tested whether the overexpression of a gene that may have a role in non-host resistance in one species could also then contribute or lead to a gain of non-host resistance in another plant species. And all these approaches identified a number of genes and components that apparently contribute to non-host resistance. And these were then often called non-host genes. That is, genes that affect non-host resistance upon overexpression or mutation. But we argue that in all instances that we could find, these also have a role in other types of immunity. And thus, the term non-host genes is misleading. And one should rather call them immunity genes. They have a role in immunity, and this type of immunity is successful against one type of pathogen, but not against another type of pathogen. This is the way one should look at it. And these genes that have been identified and these processes, these genes are involved, are typically taxon-specific and represent immunity bottlenecks in individual plant microbe interactions. So they block one particular step in the long way of pathogenesis a pathogen has to take. And usually, as said, these genes are specific for one plant microbe interaction. An exception is possibly, or are possibly, genes that encode components involved in the biosynthesis and extrusion of toxic cargo, because this was really a common theme of non-host genes that have been identified in different plant species, and which apparently play a role in multiple plant pathogen interactions. And this is illustrated on the next slide, where the outcome of genetic screen in Arabidopsis is illustrated. In Arabidopsis, the genetic search for non-host genes led to the identification of genes termed PEN1, PEN2, and PEN3. And these genes, or better, the encoded proteins, are all involved in the biosynthesis or secretion of toxic cargo at the interaction site between the pathogen, as indicated here, with an attacking fungus, and the plant that is here in this region of the cell wall. So while PEN1 is a snare protein involved in vesicle fusion, allowing secretion of toxic cargo via the vesicle transport, PEN2 is an enzyme involved in the biosynthesis of toxic cargo, and ABC PEN3 is probably the cognate transporter, an ABC transporter, which transports this toxic compound to the extracellular space. So this is where we currently are with the genetic analysis and pharmacological analysis. So we identify here and there genes that are involved in non-host interactions. But we also wondered what are novel or less explored aspects that could be investigated in the future in more detail. And we identified three areas of research. One is looking at QTL loci that could be identified from fertile crosses between susceptible and resistant genotypes in a non-host setting. But these are really rare. Why are they rare? Because usually if you have a non-host plant, there is no susceptible genotype because, well, by definition, all genotypes of this plant species are usually resistant against the pathogen. But there are some exceptions. In some plant microbe constellations, there are very rare genotypes which show a little bit of susceptibility against the pathogen, which is otherwise a non-adapted pathogen for this plant species. And then these rare susceptible genotypes can be used for crosses with the resistant genotypes, and it can be attempted to map the underlying genetic loci. A second aspect, which is in our view underexplored, is the lack of susceptibility factors. So genes that are really required for the pathogen for successful pathogenesis. And I give two examples here. One is an example of MLO genes, which, as you may know, are required for powdery mildew pathogenesis, and also genes encoding for eukaryotic translation initiation factors, which are required for puttivirus infection. And the lack of these genes in the respective plant species then give broad spectrum resistance against these pathogens. And the third aspect is the so far unknown role of microbiota to non-host resistance. So far, non-host resistance has, to my knowledge, always been studied in more or less natural soil settings where microbiota, root microbiota and leaf microbiota are present. And at the moment, we do not know whether these microorganisms contribute to non-host resistance in one or the other way, for example, by stimulating innate plant immunity. And to test this, we would need to check plant microbe interactions in sterile conditions, so using sterile plants which lack this microbiota to find out whether the presence of the microbiota contributes to non-host resistance. So these are aspects for the future to study. How does non-host resistance work? So we may identify new genes via this approach. We will learn more about the role of susceptibility factors in different plant microbe interactions and also study the role of microbiota hereby. So at this point, I stop for my part and I will hand over to Matt to continue with the next 5 questions. Matt, your turn.
Great. Thanks, Ralph. Hello, everyone. I'm Matt Moscow. Ralph already introduced me. I'm a group leader at the Sainsbury Lab. So I'll start with question 6. Do nucleotide binding leucine-rich repeat proteins play a role in non-host resistance? So this is, of course, a prolific gene family in plants. We having the nucleotide binding to ATP or ADP, and then in a C-terminal leucine-rich repeat domain, and are the most common cytoplasmic immune receptors, and many R genes that have been cloned are encoded by this same class. We know that these proteins function through recognizing effectors, either through a direct response. I can switch to a laser pointer. So we have the direct recognition where the LRR specifically binds to the effector, often in the leucine-rich repeat region, to the corresponding effector in the pathogen. There's also the guard-based model wherein the NLR recognizes the molecular status of a host protein. That's the target of the effector. And through some modification of that, whether it's through some phosphorylation event or degradation, leads to that recognition and subsequent signaling that leads to resistance. And of course, a recent model is the integrated decoy or integrated domain model, where the host protein is integrated within the NLR either through an N or C terminal fusion, and in a few rare cases integrated within the MB domain, and then leads to immunity. So this is not to say, of course, that we didn't ignore PRRs, pattern recognition receptors, also just called membrane receptors. I think that their established role in non-host resistance is quite clear, but we wanted to focus on the role of NLRs instead in BLRs because the question of their role in immunity perhaps was unclear and had substantial arguments earlier in the field for the non-host resistance. So what we do know, of course, is that there's a frequent occurrence of HR, the hypersensitive response, in non-host resistance. And what was proposed in 2011 in a review by Ralph Panstruga and Paul Schultz-Liefer was a way to integrate actually the ideas behind MBLR-mediated resistance and PRR-mediated resistance shown here with the concept that NLRs may play a more substantial role in closely related plant pathogen interactions when a pathogen is more closely related to the adapted pathogen, whereas in non-adapted pathogens that are much further away from the host, host infecting pathogens, that PRRs may play a greater role. And to some extent, this has kind of panned out as an overall theme that seems to be true. But one open question really was how much of a role do MBLRs play or NLRs play in this immunity? And I think over the last few years, particularly in the last 10 years since that review came out, we've had a lot of evidence that supports that claim. But still what remains unclear, of course, is when we reference distantly related plant species, like the example that Ralph showed earlier of Arabidopsis and powdery mildew, to what extent NLRs play a role in that interaction is still perhaps not resolved in any way. But we do know is that, for example, the PIN mutants are reduced in their ability to resist mildew in that context. So we certainly know there's a role there in which that screen was originally performed. And a good example that's come out recently from Volkan Civic and Jonathan Jones's group here at the Sainsbury Lab was the identification of resistance genes to albugo candida, which is a causal agent of white rust in Brassica species. And what they demonstrated was that that NLRs were playing a role in immunity to albugo candida. And they did this by using a magic population where they could actually identify transgressive segregants that lacked the considerable resistance that's common in Arabidopsis to this pathogen. And so in the end, identifying multiple resistance genes against white rust. So this provided a pretty good example of MBLRs participating in non-host resistance to this pathogen. And simply in this case, multiple NLRs contributing to immunity. And this has been demonstrated in other systems as well, particularly in Magnaportha oryzae, where the non-host status of different lineages is due to the presence of R genes that are, in this case, MBLRs, they're conferring resistance. So next question was question 7. Do pathogen effectors play a role in non-host resistance? So certainly the fact that in the question 6 that we know that MBLRs play a role in non-host resistance, we would effectively conclude that pathogen effectors must do so as well due to their involvement. In terms of genetic screens that have been formed, we know that pathogen effectors are recognized by plants in many non-host interactions. That's often through transient expression of these effectors in different plant species. And we also know of some examples, in this case, recent work from Bart Thomas' group, that have shown that a single effector can determine the host or non-host status of a pathogen, in this case verticillium. Now, perhaps this is a good time just to mention that unifying themes is a difficult thing, of course, because each plant pathogen constellation may have a different outcome as a result of the unique interaction of those two species. So this is a good example of where a single effector can determine the status, but that is not necessarily going to be a theme at all observed in other systems, like in the previous slide, where we saw with albugo candida and Arabidopsis, where there's clearly 4 loci or more in Arabidopsis that are conferring resistance. And in the pathogen, that's still unknown how many loci are being recognized and how much variation there is in the species themselves. But of course, another good example to which has a similar result was in Fusarium oxysporum, where also a single effector determined host range. Certainly true in these two species, but we know also the opposite is true in other plant pathogen constellations. In terms of question 8, how does non-host resistance contribute to the size of a pathogen's host range? This is central, of course, because we know the definition of itself of non-host resistance is understanding the various plant pathogen interactions that are successful versus that are not successful. We know that most plants are resistant to most diseases. And how is this related to the pathogen itself? So if we think in terms of coevolution of host and pathogen, we know that the various effectors in MBLRRs contribute together in which to determine host status. And probably the best system by which this has been demonstrated and which was referenced in the original paper by Paul and Ralph is the case of Magnaportha rhizae and also barley powdery mildew, or the mildews as well in the cereals, where these host-adapted lineages do appear to be determined often by effectors and that are also then recognized by different species, whether it's the case of wheat blast and being derived from a Lolium lineage and through subsequent breakdown of resistance in wheat, or alternatively in mildew, where a few effectors determine the host status of triticale, as the example that Ralph raised earlier. So we've seen this, at least in terms of closely adapted plant pathogen species, that this has occurred over and over again. Now, to what extent, for example, MBLRs play a role in other plant pathogen constellations, such as botrytis, is not really clear at all, and it's likely different mechanisms are underlying that interaction. But to say a species-wide distribution of these armories of MBLRRs and effectors, and likely extending also to PRRs, may contribute to this as well, as has been seen in other systems, such as in barley and its interaction with leaf rust from the work of Simone Kratzinger, published in Nature Plants last year. So having established, of course, that we know that effectors and MBLRRs and PRRs play a role, we move on to probably the question that really has motivated the field from the start, and this is question 9. So can we transfer aspects of non-host resistance to other plant species? And the answer is yes, we can. So I'll just reflect on the fact that this motivated many people, because when they look and see that most plants are resistant to most pathogens, the simple question is then how can we transfer it, that resistance to susceptible plant species to that pathogen? And this is particularly important in plant species where there might not be existing resistance to a given pathogen that exists on that plant, or where they've exhausted existing resistance within the primary and secondary gene pools. So one great example that came from Cyril Zipfel after cloning EFR in Arabidopsis that recognizes the MAMP EFTU, they were able to show that transfer to tomato from Arabidopsis conferred resistance. And another great example comes from soybean. This is from Casper Lagenbach, and using a pretty smart approach initially to identify genes that would be involved in non-host resistance by being able to disarm the initial synthesis of these toxic compounds and looking for genes that are underlying after that form of resistance. They're able to transfer them into soybean to confer resistance to fungal pathogens. And we have more examples in the review that you can look at, including transfer of MBLRRs from one species to another to confer resistance, such as RX01 in maize, Zhao et al., and then also the example of CCRPP1 from Kajanas Kajan into soybean for against Asian soybean rust from the work of Jonathan Jones and Peter Van Essa and Cynthia Kawashima. Sergio Bromachenko. Okay, so the answer was yes, and that's great. I mean, because really that's a huge opportunity for the future in terms of being able to transfer these genes between species and raises really the opportunity to engineer resistance. So moving on to question 10, and this is really when Ralph and I had a reflective exercise in terms of what else can we do in this field that we haven't done before, and in terms of expanding our understanding of non-host resistance. So the first thought was really the opportunity to do more forward genetic screens. Now, it's unclear how successful these will be, but of course, many of the initial genetic screens were performed in Arabidopsis. And Arabidopsis is a single species and a great species when it comes to doing reverse genetics and forward genetics. But of course, it's only one species. And due to the fact that there might be genetic redundancy in a specific gene family, in Arabidopsis, as we move to other species and perform similar genetic screens, we may uncover additional genes that may be involved. Now, the fact that this will, of course, uncover components of host resistance as well, because the two are intimately linked and inherently the same, isn't a problem, because what this gives us an opportunity to do is identify new genes that we may not uncover through other approaches. So for example, if we were to take a model grass species such as maize or barley and start to characterize them in detail, we might identify additional components. And so there's opportunity there in which to perform these assays. In terms of transient expression of effectors, this is a relatively new approach and has already been used to some degree, but really has more scope in the future of expressing effectors from one plant pathogen in a non-host plant species and seeing when we have or do not have recognition. The opportunity for this in particular is, of course, the utility of Nicotian benthamiana has been great, but there might be additional plant species that if we can develop the appropriate transient expression system, we might be able to screen effectors in that context as well and see to what extent there's recognition. Certainly, newer technologies such as Cas9 can facilitate high throughput reverse genetic screens, and this could occur both within the context of the plant or the pathogen, particularly when we think about complex effector families or large groups of effectors. So this is a useful approach for the future to consider. For example, there was already, not using exactly Cas9, but I mean, in bacteria, of course, as amenable systems for modification, there's already been substantial work at removing huge groups of effectors in bacteria and seeing the effect it has on virulence, and then adding them back sequentially or through chimeric composition has been extremely useful at understanding infection processes and what is being recognized or not recognized and required for virulence. So there's a huge scope here, and particularly in eukaryotic pathogens in which to take these approaches and use them. Microbiota, which has already shown up in some of the questions and which is something that is, of course, an emerging theme within the field. We don't fully understand the contribution of microbiota and its role in, one could say, both host resistance as well as non-host resistance. There's a great paper that just came out recently in Nature Plants this week describing a microbe that's in rice that confers resistance to a fungal pathogen. So sorry, already great example of an identification of a microbe that's contributing, in this case, to host resistance. But otherwise, you might have believed it was contributing somehow to a non-host resistance process to that pathogen. But there's also additional experiments that can be performed in terms of germ-free plants and synthetic microbial communities and their contribution to either resistance or virulence of what would normally be considered a non-host pathogen. So there's considerable scope to develop this in the future. And last is the idea of cryptic plant infections. So, if we think about how cheap sequencing is going to become, and as bioinformatic tools become more available, we'll be in a position where we'll be able to start sequencing field samples from diverse plants and throughout the world. And in doing so, we're likely to identify cryptic interactions of microbes that we can't normally cultivate or which to science have not been discovered previously. So there's a huge scope to really start characterizing this in an ecological context, what represents the true host range of plant pathogens in potentially their natural environment. So this will allow us to expand into non-crop systems, which have often dominated the field just due to their importance in agriculture and humans. So that's something to look forward to in the future, because that's really going to be a major game changer in our characterization of the true concept of host range. So I'll just have the concluding remarks, which are going to really point at what Ralph already showed in the earlier slides. but to bookend the presentation, so non-host resistance is a phenomenological term and not a mechanistic term. I think I said that right the first time. There you go. One of those words that's great to write, but not to say. So the fact that it describes a phenomenon is critical, because that's what confuses the field. And it's very frustrating at times. Myself, when I grappled with this, and writing this review for me was also an extremely productive process, because from the start, Ralph and I agreed on this, and it made it much easier to write the review and in which to see the entire field in a very clear way. What's striking is this term is pretty much absent from the field of animal and human immunity. And that's in part the fact they tend to treat the concept of immunity as a more comprehensive way, whereas in the plant community, it has been treated often as a strict demarcation, a very black and white result. And the main take home we're trying to convey here is that it's certainly more complicated than just concluding that something is a host or a non-host, and that there's an effective infection degree that may occur in any given constellation of an interaction. So this discrimination, as we say here, has created considerable confusion in the field, and creating terms such as non-host resistant or non-host genes or non-host resistance genes. And that really has to be avoided in the future, because that removes the fact that they're likely participating also in host resistance, especially when we start to consider why those genes exist in the first place and how evolution selects for their presence. It's likely they're participating in host resistance, and that's overlaid in its role in also conferring resistance to non-host pathogens. And last, we hope in the future that in the field, people will adopt a more holistic perspective on plant immunity, and use this term more carefully and really in its original context as a phenomenon, because it will help to make things clearer at the end of the day for everyone when reading papers and understanding and trying to build additional models to explain function. Right, and I think with that we have questions, in which I already know looks like there's 17 questions in the Q&A, which Ralph and I can begin to address.
Well, thank you very much for a terrific talk, Matthew and Ralph. I really liked the collaborative approach that you took to writing this, and you can see that's reflected in this terrific talk. And also that you took the time to really shape how you think about this, and that we can moving forward. The top 10 was really an effort as a community to identify these key unanswered questions, and we hope is an entry point for new scientists to this field. But it was also to stimulate us to revisit them and for us to brainstorm. And that's been an incredibly positive experience for me, and I think for others. And Matthew referenced that when he talked about the brainstorming that he and Ralph did to think about where to go. Like once we've defined some of these big questions, what are the ways we should be looking at it now? The world has changed. We have tools that we didn't even imagine 5 years ago. And how can this change how we approach things? Okay, so just a reminder to type into the Q&A box. I see several of you have figured that out. And so I'm going to start by reading them. And there's a couple of people who, because of time change issues, show up at the bottom, even though they were early. So I'm going to start with a very general question. Who came up with the original definition? So either, yeah.
I think, to the best of my knowledge, it was Michelle Heath, Canadian plant pathogen researcher in the early 1980s. If someone in the audience knows better than me, he or she may shout out, but I think it was Michelle Heath, maybe end of 70s even.
1977.
1977.
Yeah. But I don't know if in that original paper, if she defined also non-host resistance as resistance to all strains of a pathogen by all accessions of a species, a plant species. So that specific definition, I don't know when that emerged. Off the top of my head, I can't answer that.
Yeah, maybe it was put forward by Hans Toller Christensen.
Well, I know that Rinsnix had it certainly in the late 80s. He introduced that concept.
That was then much earlier,
yeah. But in the late, that was like 87, 88, he had these Euphytica papers. But I suspect it probably was introduced earlier than that. And I'm sure by Michelle Heath or others.
I think this does raise an interesting point for me as an outsider to this field, is that the way you frame a topic or an idea really affects the kinds of questions that you ask. And so that's why I really like this reframing to a more mechanistic view that Ralph and Matthew have done. Okay, moving on to another question. Montserrat Lopez Coria says, the shift between host scenarios must be sequential, or is it possible a shift of non-host to host without a pass for intermediate states? Well,
of course, when we detect it as a shift, we have missed probably the intermediates. It is likely to assume that in many instances, a shift will be sequential. But as in the part of Matt, it was pointed out, there are scenarios where the loss or gain of a single gene can be the matchmaker. And then all of a sudden, there might be a quite dramatic shift.
Okay, I'm going to move on to one by Ignacio Eduardo Maldonado. And I'm sorry, I can't read the second half of your name because it goes out of the box. But Ignacio asks, how about interaction with other members of the microbiota, which could change from a host scenario to a more intermediate host or non-host scenario? And this ties into the point that both, I think, Ralph and Matthew raised in their talks, their part.
I think this is an emerging area to study. And of course, that's the goal with any microbiota-based discovery pipeline where you're trying to engineer immunity, would be to try to transition from a host context to a non-host context via the presence of that microbe. Certainly from a biotech strategy, that's a major goal. So I think this is something that we'll learn more about over the next 10 years in terms of discovery, both from the academic and private sectors, I anticipate. I
agree. I think that's a really exciting area.
And this recent paper in Rice is a great example. The Matsumoto published on the 4th of January this year for this Burkholderia resistance that's determined by a bacterial species that's a endophyte. So I think that's already a great example of where that came. It just came up in my Twitter feed, so that was fun to discover.
Oh, and Matthew, I'm glad you mentioned that, the Twitter, because that reminds me that Morgan Carter, one of our new assistant features editors, has taken over the MPMI Journal website today, and she is live tweeting this talk and the Q&A. So thanks very much, Morgan. And so if people missed some of it, you can always check Twitter to get the Cliff Notes version. Okay, so next question. How would stacking of NHR genes affect the plant due to their associated original functions? And this is considering the ability of broad spectrum recognition and frequent activation. I think the real focus there is the stacking of NHR genes. So sort of several steps forward, since you've just said that NHR genes should really be considered immunity genes.
Yeah, I guess the pleiotropic effect of any gene always has to be considered based on the original function. This was a concern with susceptibility genes, whether they'll have a pleiotropic effect on plant development. Certainly MLO is an example of this. In fact, of the mutations in MLO, only a subset are routinely used in agriculture, in barley at least, due to the fact that they do have a pleiotropic effect on the plant. In terms of genes that may be participating in non-host resistance, I think it's a case-by-case basis on whether they are going to have a pleiotropic effect on the plant. Certainly in the case of MBLRRs, In the field, there's a perspective that perhaps MBLRs may have a negative impact on plant fitness, but there are now several cases of where they clearly don't. I mentioned in my part of the talk, CCRPP1, which when transferred from Cajanus cajan to soybean, didn't seem to have any impact on plant fitness. Whereas of course, there are other cases where we know that is the case when an MBLR is transferred. Careful selection of promoter must be done, or else it may lead to stunting phenotype. Many of those cases seem to also be involved with a guard-based model of recognition, where it can lead to a kind of a constitutive active response to the presence of the guardee for that NLR. So because often its pleiotropic effect on plant development is determined by its molecular function, I'll go back to the first thing I said, which is it's a case-by-case basis. One has to be careful in considering what they use and how they use it, and tune it appropriate for the system that they're going to use it for, particularly in engineering immunity.
Okay, next question is by Aranka Vandenburg, and they ask, as all various kinds of molecular mechanisms are underlying NHR, what is the function of using the term NHR, which is something that you've certainly, an issue you raise.
Yeah, as we pointed out, we would argue to ban the term NHR because it's so misleading, and better talk about effective immunity instead of NHR. So I think Matt nicely summarized that at the end and also in the concluding remarks.
Okay, next question is by Jyotsna Karanam, who asks, are there any examples where host extension jumps occurred earlier? And I'm not sure it's earlier relative to what?
Earlier than what?
Maybe, maybe it's sort of seeing, maybe this is reflecting a question, sort of seeing the intermediate steps. Are there any cases where it looks like a huge jump, but there were some intermediate?
We can assume that due to evolution, host range extensions and host jumps occurred all the time, also in the past. It's of course difficult to validate this because that would need very good fossil records or something like that. It's really difficult to prove what happened in the past. Yeah. But based on the amount of the number of host shifts and host jumps we see nowadays, there's no reason to believe there was significantly less in the past.
There is the one clearest example of a major host jump was from an animal pathogen onto plants, and that was the ancestor of ergot and so claviceps and epicoliae, which was recently discovered to have a horizontal gene transfer into grasses actually for fusarium resistance. But those two lineages actually come from an insect infecting fungal pathogen. But that happened like 100 million years ago. There was that host jump. So I think one thing that's lacking is just enough sequencing data and enough species, because often these could be cryptic host jumps and we won't know until it's too late that it's occurred. So we're really at this turning point where we're going to have the genome sequence for many of these species in which we'll really be able to make strong conclusions. So it'll be exciting, looking forward when we have that information to be able to make those conclusions, whether this has been a recent host jump or not.
Yeah, that's amazing. The rapidity of sequencing right now. Yusuf Hamidi touches on this idea of conditional non-host resistance that you raised, that Ralph raised. How does the environment factor, G by E, influence the non-host resistance?
As far as I know, there are not many or not any detailed studies on this aspect. It is just at the moment that we see that environmental factors can influence non-host resistance. And yeah, we simply need further analysis to dig deeper into that.
I'm really glad somebody brought up this question, because the next focus issue that MPMI is going to have coming out, not a year from now, so next January, so start writing, is how does abiotic, it was question number 2, top 10 question number 2, how does abiotic stress or the abiotic environment influence plant microbe interactions? And it would be fascinating to see even just a reviewer perspective talking about this issue of how does the environment affect non-host resistance, but also we're looking very broadly. So please, you'll see that announcement coming out pretty soon. And I should point out that this top 10, this whole brainstorming that we did, this was the result of lots of conversations and talking and people raising the issues in seminars and really coalescing, a lot of discussion. What are the big questions? And non-host resistance. You can see all the open questions here. This is an area that's really open for lots of questions, lots of interesting approaches. So I hope that younger scientists who are following this take note. Okay. Montserrat Lopez Coria has one more question. The phenomenon of non-host resistance, could it be similar in the interaction of plants with a beneficial fungus that cannot stabilize the healthy interactions? So again, this is really asking about the interplay of plant microbe interactions, perhaps the way mycorrhizae sometimes provide some sort of systemic resistance.
I still try to understand the question fully, I have to say.
So I guess if I were to, I mean, we can talk it through, Ralph. So basically expanding upon, well, what we would say is non-host resistance in the context of a pathogenic microbe, expanding that concept to sort of non-host interaction with a beneficial microbe.
All right.
Which yes, I would argue, I mean, once again, similar to how it was mentioned that in a plant pathogen constellation, how there's a huge number of interactions that can occur between diverse fungal, bacterial, fungal, et cetera, pathogens and a different plant species. It's the same thing with beneficial microbes. They have to often achieve many of the same steps that a pathogenic microbe will achieve, but at the same time, communicate in an appropriate way to which to let the plant know that they're actually a beneficial microbe that's going to be let in and facilitate transfer of nutrients and other beneficial outputs. So I would say that the concept itself could be expanded to both in terms of the way you look at it, in terms of an interaction and molecular dialogue between the plant and the microbe.
Yeah, I would agree. These are analogous concepts, so to say, for a pathogen and for a beneficial microbe or fungus. So as Matt said, multiple steps are required, and at each step it can fail. And yeah, so this is really analogous to.
Yeah, and here you have this idea
suggest.
of pathogen, of beneficial, and of course that huge in-between area of no interaction, which includes non-host resistance, as well as other commensals, and that's a huge question.
Yeah.
So
there's also the examples of the transfer from, sorry, there's also the cases where they switch from a beneficial to a pathogenic state,
yes.
After sometimes only the single loss of a single gene, or a environmental dependence for that to occur,
or vice versa, right?
Yeah.
The reversal from pathogen to can be with a single loss. Okay, so Sophia Maheka Garnica asks, can NBLRR also detect PAMPs, or do they detect effectors exclusively?
So the cool part about the NOD, sort of the NLRs of animals, is that they do recognize PAMPs in animals, particularly in humans, for flagellin recognition. But that's also due to the fact that they're brought in vesicles and then recognized. So it's a fascinating phenomenon there. I guess I would say it remains to be seen of an example, if that's fair, Ralph.
I do not know of any plant example. As you correctly point out, in human beings, there's about 10 NBLR proteins. They recognize different types of MAMPs and PAMPs, flagellin, double-stranded RNA, and so on. But I do not know of an example from the plant field.
Yeah.
Only the opposite, that a plasma membrane resident receptor recognizes an effector. There are examples for that.
Yes, yeah. I guess the problem is that when we think about what makes up a MAMP, I wouldn't think of them as much as PAMPs as MAMPs. MAMPs often are accidentally released by the microbe. EFTU, for example, is a part of the elongation factor inside of bacteria. So that's actually in the bacterium. And you have flagellin, which of course is a part of the motility of bacteria. So, and then we also have things like chitin as well, which is part of the cell wall of fungi. So those get shed from the bacteria or fungal microbes, and then those are recognized at the plasma membrane in the apoplastic space. So the question is always, if NLRs were recognizing some MAMP, it would have to be brought into the cell. And one of the reasons why likely we won't discover that is because nothing's actually making its way into the cytoplasm where the MBLRs reside.
At least not early and not in huge amounts. So if at all, yeah.
So Allie McLean posed the question, I'm intrigued by the idea of susceptibility genes being associated with NHR. I'm familiar with the concept that susceptibility genes are associated with disease by definition, but had always and only associated resistance or defense-associated pathways with NHR. Can Ralph comment further on this and how important a role he thinks susceptibility genes may play in the outcome of NHR? And she also compliments both of you on the seminar, and Matt, particularly on your beard.
Thank you, Ali.
Well, for these susceptibility genes, well, there are two sides of the same coin. So if they are present, they confer susceptibility. If they are absent, it results in resistance. And this type of resistance often looks like non-host resistance, as in the case, let's say, my famous example of MLO-mediated resistance. If the MLO gene is lacking, then the fungus fails to enter the host cell like a non-host, non-adapted powdery mildew pathogen. Apparently, this gene or this protein is really required to overcome immunity, non-host immunity. And so these are the same gene or protein can do both in a way, if you wish. It can confer susceptibility, and in its absence, it results in a great and a very durable and broad spectrum resistance. So yeah, I think there's nothing I could add here. Matt, do you see?
No, I guess linking that question to MLO, I mean, there is some work that's identified additional susceptibility genes, but they haven't been, they haven't been as easily identified, comparatively speaking, as R genes, for example, in natural variation. But it's a great comment to what extent certain genes are essential for susceptibility, and that loss of those or impairment of those in some ways lead to non-host resistance. I guess the difficulty in their identification has been, if they do exist, in many cases they may be essential for the plant's development, in which case they are not discovered through any forward genetic screening to date. Even MLO, of course, it has, other members have other roles, for example, in pollen tube and in fusion.
Well, at least the examples where we do see a clear effect are very rare. So there might be others which have a more subtle effect, not so dramatic, but the very strong examples are rare. Yeah. And this, as you correctly point out, might be due to the fact that these genes have other roles or are redundant.
Yeah, we discussed this at length among ourselves with Ralph and I, was the role of the effector repertoire itself and individuals' effectors, contribution to fitness. This is often discussed in the community and will be covered in some of the other top 10 questions. And then those effectors themselves, how they contribute to non-host resistance, and how do they also adapt to a host, are all important questions. And we only have a few well-characterized examples. And that's something that we hope with the future will also come out through work by many in the field.
Yes, thank you. And that reminds me that we do have question 5 review already published, touching on some of these ideas of effectors and PAMPs, and maybe Ashley could type that link into the chat so everybody can see it. And moving on, Sham Solanki asks, I wonder if the specificity of effector triggered NLR, NHR translate into a pathogen species specific NHR response.
Yes. Wait, to make sure, I'll reiterate, I'll rephrase the question to make sure that I understand it, and then Ralph can confirm that I understand what I'm talking about. It's the idea that, of course, any plant may look resistant. It might be green, but underneath that are a pathogen-specific response that leads to its immunity. So the idea that you could have, you know, an effector-based recognition, 1 or 2, perhaps, maybe more, and that the plant looks immune. But to a different pathogen, the same thing could occur and it looks immune. So both are called non-host resistance, but in fact, all have their own individually tuned immune response to the pathogen. But I would say yes to that. And that's just similar to what we see as host resistance, which is sort of the theme of this article, to say that the two are inherently intertwined. I think that in the field of immunity of plant microbe interaction in general, of course, our outputs are on the 2 extremes, resistance and susceptibility. So because we have those as outputs, inherently, the response is always the same. It's just that the process by which that may occur may be very specific to the interaction.
Yeah, so the next question is about abiotic stresses, and I'm just going to reiterate that that's a huge area. How does abiotic environment affect plant microbe interactions? Really exciting area, lots of unanswered questions there, including with non-host resistance. So we're going to move on to a question from Dennis Halterman. He says, great talk and paper. And I should remind you, if you guys want all the details, you should dive into the paper. It's got an excellent discussion of some of these ideas. Dennis says, I like your reasoning and agree that it would be nice to get away from the non-host label. Are there any pathogenic microbes that are completely non-pathogenic on plants, such as animal pathogens? And can non-host interactions be better defined as non-plant pathogens, such as E. coli, Yersinia, that can enter plant tissue but never cause disease on any plant? For example, what are the differences between animal pathogenic Pseudomonas versus phytopathogenic Pseudomonas?
I would argue it's hard to say that there are microorganisms that never cause disease on any plant, because this would require really to study every individual interaction with every plant species on this planet. So we have to consider that even non-pathogenic microbes probably accidentally release MAMPs or PAMPs that are recognized by the plants and trigger an immune response. So it's a bit difficult to discriminate whether they are non-pathogenic because they do not have a pathogenic program, or whether they are non-pathogenic because the plant responds to the MAMPs maybe. So yes, it's a little difficult to make a very general statement here.
Yeah.
So there are animal pathogens which also can cause, or at least some kind of plant disease that are a little bit pathogenic on plants as well.
Or or can at least survive long enough to then cause disease in humans if it
Yeah. And I think also some of the human stuff, often when they are sequencing stuff out of, for example, diseased lungs, they come across things like rhizobia, which we normally associate with plants. And you know, again, you have these sort of chance interactions that happen in the environment. And maybe if you are diseased in some other way, they can get in and do what they like to do best. So yeah, it's a very interesting question. And certainly there's, I'm spacing on the name, but there's beautiful examples from the symbiosis literature. Oh, Brucella. Brucella abortus is an animal pathogen, and one gene required there for persistence in the animal is also required for persistence in of rhizobium in legumes. So I do wonder, you know, once bacteria get together in the environment, they're swapping information back and forth with these via conjugation. How rapidly can things move? Okay, we're getting up to 12:30, so I am going to take, let's see. Do either of you have a question you'd like to answer in the remaining list? There's a lot of questions. And normally we would try to go through everything, but we have had, this has been a terrific talk, terrific questions. Matthew?
Well, I can see that instead of shortening the list of questions, it ever grows.
Growing.
Well, isn't that the way it works? You ask a question, you get an answer, and you have more questions. So, yeah, it's so we have questions about. Yeah, go ahead.
Yeah, Rimco's. We've had this with others. You know, what's the utility of the term? If it's difficult to empirically test in many cases. Oh, I mean, I think it's like any model, any concept we come up with in science. It's not meant to be right or wrong. It's a way in which to frame questions. So I think there are some cases, certainly in many non-adapted pathogens that are closely related, where actually one can do experiments. So I think it's still especially useful to retain the concept of non-host resistance. And certainly the observation still, and the original concept itself from Michelle Heath, that the observation that most plants are resistant to most pathogens. I think that's just still a fascinating phenomenon in which to observe and maybe dissect, if possible, in specific systems. So I haven't given up on it yet.
I think there's still many things to ask. And yes, and Valentina Klimiuk says something similar. What should be the modern definition of non-host resistance? And that might be a good way to end.
Well, my suggestion would be effective immunity, effective plant immunity.
Mm-hmm. How about you, Matthew?
That's where we came to in our discussions with the review, was that non-host resistance is an example of an effective, efficient immune system that's not being corrupted by effectors and is mediating an immune response.
Okay, well, that is a terrific point to end on. And I hope that this has stimulated your interest in looking at the details in the paper, because it's a beautiful review. It goes into a lot of these questions. And we also have a really nice Q&A and interactions between Ashley, who is our IS-MPMI communications person, and Ralph and Matthew asking them about this. And so this Q&A and interactions is, I don't know if we can type that into the chat actually, but it's a great way, and it might also be a nice way for students to engage with these concepts and these big ideas. So, well, I'd like to thank everybody, especially those of you who have stayed until the end here. And terrific questions, great talk, top 10 questions. These are the questions that are motivating a lot of people in the field. And you can see that by the number of questions that are coming out in the Q&A, right? This is something that people are wondering about. And that durable resistance is like, it just is like something that draws you on, thinking, is there a way we could transfer durable resistance into crops? So lots of really open questions there. Okay, so I'd like to thank Matthew and Ralph again, and I hope you will join us in February. We'll be posting the speaker for that very soon. Okay.
Yeah. Also, bye-bye from my side and stay healthy, guys,
Yes.
these days.
Thank you, everyone.